Welcome, everybody. My name is Markus Habich, and as Fabienne told you already, I'm the product manager for the imaging reagents here at Miltenyi Biotec, and I'm really happy to present today's webinar to you. And at the second half of the webinar, Hussein will take over. And I would say let's directly start with the agenda of today's webinar. What do we want to have a look at? So first of all, we would like to introduce 3D imaging and address the question, why actually to image large samples in 3D? I'm going to focus on the sample preparation for this 3D imaging workflow. And then Hussein will take over and present to you the large-scale 3D fluorescence imaging solution that we provide, and also show you which applications this can be used in the research areas of immuno-oncology and neuroscience. So then I would say I would directly start with something that I guess all of us know, and this is conventional 2D analysis of tissue sections. This is in combination with antibodies, also referred to as immunohistochemistry. And this is a gold standard method. It has been around for over a century, and we know these images from many publications. So here you can see tissue sections being stained with dyes or antibodies in a chromogenic detection manner. Or here on the right-hand side, you can also combine this with immunofluorescent microscopy. But first, I would like to take a step back and ask the question, why actually do we do the 2D imaging of sections? And this has to do with that large samples, such as here in this example, the onion, light cannot pass through them, and this makes them opaque, right? This is what we all learned. If you take a thinner layer of the onion and remove this monolayer of cells here, you can now see through the sample. And by decreasing the sample thickness, you now allow light to pass through the sample, which makes it translucent. This is the basic principle how microscopy of thin samples, for example, tissue sections, works, right? So if we have a look at the workflow to analyze these tissue sections in 2D, you generally start out with a rather large sample. Here described as a mouse brain, for example. And what you do is you perform physical sectioning. So you section it in thin slices, and then these slices, they can be, for example, stained with antibodies to label these structures of interest, and they are then imaged in a standard microscopy setup, for example, a confocal microscope or a wide field microscope. And what you would obtain is here this 2D image, as you see on the right-hand side. This is a mouse brain section stained with a CD31 antibody, which is labeling endothelial cells and which is often used as a blood vessel marker. What is great about this workflow is that it allows the microscopy of opaque tissues that we could normally not analyze. This was the example of the onion I showed before. The imaging of these tissue sections is straightforward, and since it has been an established standard method, a lot of protocols are out there, a lot of optimization has been done, and yeah, it's basically performed in every lab, very straightforward. However, if you would like to obtain more information, especially in 3D, this tissue sectioning workflow limits you. And at first, you compromise, of course, your sample morphology by sectioning. And this can introduce artifacts which you observe in your microscope, which are actually not present in the sample. Also, 3D information obtained by tissue sections can be very poor. On one hand, if only a partial sampling took place, so only a few sections of a large sample have been analyzed, there is few 3D information, but even if you obtain many sections, the alignment later in the image analysis is not very easy and often complicated to perform. Of course, if you want to section the entire sample and stain it completely, this is connected with a very high manual workload to analyze these sections all individually. And finally, probably most importantly, for the biological research question, there is a big potential to miss information by not analyzing the entire tissue. For example, if you have a biological specimen and would like to analyze for cancer cells, then there is the risk that you miss some metastatic cancer cells if you're not sampling the entire tissue. So how can you overcome this? And what if you could actually analyze the entire sample that you do not section in a microscope? And this is basically what the 3D imaging of large cleared samples is addressing. And with this technique, you're now able to see the truly full picture. And this is here an example by Dr. Ali Ertürk, who was able to clear an entire mouse, and with labeling, he was able to see here nerve structures in the context of muscle cells and other organs, which would have been never possible without this 3D imaging workflow technique. Then we would kickstart with the basics of tissue clearing and have a look why tissues are actually opaque. And I would like to start out with this little kitchen experiment here we did. And these are 2 glasses filled with either distilled water or distilled water with a drop of milk. If you first focus on the left glass of water where we shine a laser pointer through, you see the light only at the interfaces of the glass to water refracting, but you do not see any light in the distilled water, right? And this drastically changes as soon as you put a little drop of milk inside the distilled water. So what is happening here actually? And basically the opaque tissues, they are actually formed by molecules that have different refractive indices, as we call it. And this causes high light scattering, and which leads us in turn to lack of transparency. This is visualized here in this very simplified model. We have here an opaque tissue, such as this human ovarian tumor you see. Incident light is shining on it, and there it hits a lot of different molecules. So can hit water molecules, proteins, lipids, and they all have different refractive indices. And this causes light scattering, refraction, and this in the end leads to the fact that the light cannot shine through because it's scattered through the whole tissue. And tissue clearing has the aim to reduce this opacity by unifying the refractive index of these opaque tissues, either by removing, replacing, or modifying the tissue composition via chemical reagents. Importantly, you have to preserve the structure for this. So in this example, what you see, the proteins, they would stay at their place because those are the ones that should be analyzed, but the rest of the tissue has been matched with a refractive index to these proteins, and the incident light can shine mostly through. And this can be visualized here in this human ovarian tumor. You can now see the grid below it. It's now translucent, and this is the prerequisite to analyze a large sample in 3D. So this is great, this basic knowledge, but probably some of you also have faced this. If you want to get started with tissue clearing, what you will find in literature are more than 30 clearing protocols that have been described so far. Principally, they can be classified in 4 different families: the solvent-based family, and very famous representatives are members of the DISCO protocols. There are also simple immersion techniques, hyperhydration, cubic is one of the most famous hyperhydration protocol, but also hydrogel embedding. Clarity is very famous here. But if you see all these different protocols, the questions arise, okay, which method should I actually start with? And this is often depending on the application. And once you decided for a certain tissue clearing protocol, often you have to optimize it for your organ. And this can be very time consuming. And if you choose, for example, solvent-based methods, which are very efficient often and offer very quick tissue clearing, a pain point is that it often includes toxic components and cannot be handled easily in every lab. So you see, as a beginner, there are certain challenges that are faced. And this is the point where I would like to introduce to you a simple 3D imaging workflow where you're guided throughout the entire steps. So in here, this is a simplified workflow that shows you the immunostaining, which we support with antibodies that have been validated for this 3D application. Then the tissue clearing step, we offer you a product which is called the Max Clearing Kit. And finally, the analysis of the sample in 3D can be performed with the UltraMicroscope Blaze in combination with the Max Imaging solution. So this is a complete workflow that we offer here that we would like to explain to you a little more in detail. Because we talked about tissue clearing before, I would start out here with the Max Clearing Kit and explain to you what this kit consists of. As mentioned, this is designed to be simple, and in addition, it is non-toxic. This is a solvent-based clearing protocol consisting of 3 different components. It is a permeabilization solution to make the sample accessible for antibodies, which is then used with the antibody staining solution, and then finally the clearing solution. And what this clearing kit can accomplish is shown here with these 2 exemplary tissues. So neuroscientists would, of course, analyze mouse brain, and this is a hemisphere that you can see here that has been nicely cleared and can be optimally analyzed via 3D imaging, but also for oncology or immuno-oncology applications, such a human ovarian tumor can be nicely rendered transparent for further analysis. So we have here a efficient and a robust tissue clearing. I'll show you in a second that you're not only limited to these kind of organs. It is non-toxic and solvent-based. And since it's solvent-based, it is very quick. And since it's non-toxic, you don't have this pain point. This can be applied in any sort of lab. And we optimized it for the whole mount immunostaining, so labeling with antibodies. And to make it as easy as possible, we published here protocols on our website. This you can check out. You also find a nice video how to stain a mouse and clear a mouse brain hemisphere. But we also offer protocols to clear and stain human cerebral organoids or primary tumors and xenografts. What I had mentioned before is that you're not limited to mouse brain hemispheres or tumors. With this clearing kit, you can clear a broad range of tissues, and this is seen here. So as a matter if you use mouse lymph nodes, lung, gut, or even a paw here, these are nicely cleared and rendered transparent in a very efficient manner. Okay, so after presenting the tissue clearing option of Miltenyi Biotec, I would like to focus a little bit on the immunostaining aspect of these large samples. And these antibodies we will release soon. And first of all, having a look what is so special about antibodies. I mean, all of us know the 2D immunofluorescence or immunohistochemistry staining of samples. It is easy to stain a 5-micron section, for example, of an FFPE fixed tissue. But if you look at this 3D imaging approach, you're now talking about a mouse brain hemisphere, which is 120 times thicker as a sample. And just to show you what this means actually is here, this is the thickness of an FFPE section, 5 microns in comparison to the mouse brain hemisphere. So I think it's easily understandable that this is not straightforward and comes with certain challenges. And it's not only that you have now this large sample, but also you have to make it optically transparent, right, with tissue clearing. And this is another aspect that has to be accounted for, that the antibody staining really survives also the tissue clearing that follows afterwards. So many beginners face these huge problems that the antibodies do not penetrate the tissues properly. It takes very long to stain these large samples with antibodies, and also these large samples come with a lot of unspecific signals, so background, making this whole process very cumbersome and also expensive to validate these antibodies. And this is something we were focusing on when validating a new portfolio of antibodies for this application. Just to show you what this actually means and what you might have encountered yourself as well, is when staining large samples with antibodies that are non-validated or non-optimized, you see here often a rim that is caused by antibodies not penetrating the tissue properly. This is here a cerebral organoid of human origin that has been stained with a beta-tubulin 3 antibody. This is a neuronal marker, and you see that this is not acceptable as an antibody staining of a large sample. And the difference is that if you have validated an antibody, it is working for this application, and you know the optimal concentration and it has been optimized, you see that the antibody fully penetrates the sample. And this is simply the work that you can save when using validated reagents. And for this, we have our antibody brand called Reaffinity. Those are recombinant antibodies that are conjugated directly to very bright and photostable fluorochromes of the biodye family. This is now an example with a brain organoid, but we do this with different tissues of increasing size. So the largest 3D sample are the cerebral organoids. They are 1 millimeter in size, which is already a very thick tissue section if you go for 3D imaging. And it's definitely considered as a large sample. And you can see this here with these markers that we stained it with, showing a nice penetration inside the tissue. But we also use human tumors here as an example with the pan-cytokeratin antibody, but also going up to large samples such as mouse brain hemispheres, which you can see here. This is one image of a CD31 staining, and you can appreciate that the antibody nicely penetrates throughout the entire mouse brain hemisphere. There is one thing that I would like to mention as well. When doing direct immunostaining, you of course have the advantage that you are very quick in the protocol. And one thing that these large antibody stainings or these whole workflow protocols have in common, if you use large samples such as a mouse brain, is that the protocol in total is pretty long. And most of the time is accounted for the antibody staining. And what you can see here in this indirect immunostaining, you first label with a primary pure antibody, and then you detect this one with a secondary antibody. And this incubation step is equally long as the primary incubation. And this is an example for a mouse brain hemisphere. You end up with a 20-day protocol. So it takes you 3 weeks to acquire the data finally. And this changes if you use a direct immunostaining approach, which with fluorochrome labeled antibodies, you cut this down by 50% of staining time, which means that you can analyze your samples 7 days earlier. Of course, if you use a direct immunostaining approach, you need to make sure that the signal is also good enough that you obtain. And this is shown here in this exemplary experiment. So what we did here is we used a mouse cerebellum and we stained it either with a fluorochrome conjugated antibody in a direct immunostaining approach, or we used a secondary antibody in a indirect immunostaining approach. And what you can see here and appreciate is the very crisp image that you obtain with the direct immunostaining approach. So the signal to background ratio is very good. You can differentiate here nicely the Purkinje cells in the mouse cerebellum, but also the axonal tracts reaching out here. You can nicely visualize and differentiate. And this differs here in the indirect approach. You definitely get a stronger signal, but in the end, what counts is the signal to background ratio for your data analysis. And this is inferior in this example. All right. And with this, I would like to show you lastly one example what you can do with this workflow. And this is a mouse brain hemisphere being cleared with the max clearing kit and then analyzed with a CD31 antibody. And you can here differentiate the fine vasculature structure of the mouse brain that is stained with this marker. So if this would be interesting for your research, we would like to welcome you to this 3D imaging workflow and the sample preparation, which I am already at the end with my part and would like to hand over to Hussein now.
Thank you, Marcus, for that nice introduction. Hello, everyone, and thank you for joining our webinar. So after staining the sample and clearing it, we would have to image it in 3D. So what imaging method can we use here? To answer this question, we have to look at the available microscopy and imaging techniques and their capabilities. Here you can see the size range that is feasible with different techniques, starting from a single molecule that requires sub-nanometer resolution and all the way up to a whole animal, where imaging techniques provide a resolution in the millimeter or high micrometer range. But what if we want to study let's say drug targeting at the cellular level in an entire organ? How can we go about imaging an organ at high resolution without having to section it? The answer here lies in the combination of tissue clearing that Marcus presented and light sheet microscopy that unlocks huge potential and bridges the gap between all these techniques, therefore providing us large-scale imaging at cellular and even subcellular resolution. And in fact, the UltraMicroscope that I will present today was the first commercial microscope combining the advantages of light sheet imaging technology with tissue clearing. Let's have a closer look at techniques that enable 3D imaging to see why light sheet is the instrument of choice here. Typical confocal and two-photon fluorescence microscopes that are depicted here, they use the same axis for illumination and fluorescence collection. To image a certain field of view, they use point scanning to excite the sample, and then the image is reconstructed point by point using a PMT detector. Confocal microscopy achieves optical sectioning using a pinhole that rejects out-of-focus fluorescence, while two-photon microscope relies on the fact that two-photon absorption only takes place at the focal plane. In other words, you only excite what you are observing. Now, if we take a brain, we know that it's opaque and will therefore scatter the visible light. Confocal microscopy results in limited penetration depths and suffers from unnecessary photobleaching because you excite everything and you only confine the emission or the collection. Two-photon can image deeper into the brain thanks to its red excitation, but the signal will deteriorate with depths beyond 1 millimeter. Therefore, these two methods are not suitable for large-scale imaging. The method of choice for large-scale imaging is light sheet microscopy. Here, we use an excitation axis in the form of a light sheet that is perpendicular to the emission axis, as you see on the right. This way, we create a microscope with an inherent optical sectioning ability. This means we have confined excitation and emission only to the focal plane. And here it has a very large field of view as well. Moving the sample in the light sheet allows you to obtain 3D images. No-point scanning means significantly higher volume throughput, and the lower complexity of the system makes the technique more accessible to non-microscopists. Tissue clearing here unlocks the ability to image much larger samples, and the clearing transforms the microscope into a high-resolution virtual microtome, or organ and tissue scanner, if you will. Let's have a closer look at a light sheet excitation. Here you see a cylindrical lens on the left that is used to generate a light sheet that will be used to excite your sample from the side. This light sheet, as everything else, is not perfect. Therefore, as the generated light sheet diverges, it approximates a plane only over a given horizontal range called the Rayleigh length. This is the shaded area that you see on the right. This is where the light sheet is thinnest and where fluorescence detection will take place. To achieve now an appropriate illumination for a particular sample, the planar range of the light sheet has to be adjusted. Choosing a low numerical aperture of illumination results in a broad field of view at the expense of a low Z resolution. Here you can see how the light sheet thickness does not vary much across a large area. Adjusting the light beam width increases the illumination numerical aperture and therefore produces a thinner light sheet with a smaller planar region that is used for imaging. Here you get a high Z resolution at the expense of a narrow field of view, and this is suitable for imaging small samples. But what if we wanted high XZ resolution and a large field of view at the same time? The UltraMicroscope offers a solution called dynamic horizontal focusing. To that end, the Rayleigh length where the light sheet is thinnest is rapidly scanned across your sample, and a sequential series of high-resolution images are taken across a desired field of view. And then they are automatically stitched together into a single high-quality image by the software. You repeat this process for every stack, and then you get a high-quality 3D data. And in the next section, I will introduce UltraMicroscope Blaze. Here you can see the microscope, and let's start with illumination. The system has a symmetrical design at the bottom with light sheet optics that generate 6 thin light sheets, 3 coming from each side, that illuminate the sample bidirectionally. And the new advanced optics here provide overlapping light sheets for more homogeneous illumination. The emission signal is collected perpendicular to the illumination axis through our immersion MI plan objective lenses. And then the detection unit here has an automated magnification changer that allows you to switch easily between macro imaging and subcellular imaging. This is followed by a large tube lens that allow automated chromatic aberration correction, which makes the system well suited for multicolor acquisitions. And the signal is eventually collected using a highly sensitive CMOS camera. In summary, the microscope has 3 important features. It is designed for imaging large or multiple cleared samples, and the full automation makes it very user-friendly and easy to operate. It has unique light sheet illumination and detection optics, which guarantee superior image quality in all clearing media. One thing that makes the UltraMicroscope Blaze unique in the field is its large sample chamber that allows you to visualize many fine details all in their native location. With the UltraMicroscope Blaze, you can image up to an entire mouse, as you see here in this example, that reveals the neuronal connectivity map in the whole mouse body. You can also choose to mount multiple smaller samples, such as whole brain or other rodent organs. This can be highly valuable if someone wants to screen multiple organs or organoids for drug screening, for instance. This sample holder is then loaded into the imaging chamber that is filled with the imaging solution, and then with a click of a button, you can start a batch measurement without any user intervention. When it comes to full automation, in addition to the batch measurement, you can see here that the entire detection unit moves, the objectives are automatically exchanged, and the sample that you see on the right is you have an automated sample release. The automated release of the sample is not only a matter of convenience, but it also provides safety for the user, especially when working with aggressive organic solvents. And to guarantee that we remain in focus after objective lens exchange or sample exchanges, we have the autofocus feature. All this leads to an easier handling and reduces the hands-on time for the user. Let's have a closer look at the MI-Plan objective lenses that are designed specifically for light sheet imaging in immersion media. As you can see in this table, we have objectives covering a range from 1.1x up to 12x. And when combined with dipping caps, they provide distortion-free imaging in all clearing media. The dipping cap protects the objective and provides the refractive index matching needed for high-quality distortion-free imaging. In addition to their superior performance, they provide maximum flexibility. And combined with the automated magnification changer, you can now image at the macro scale, such as the vasculatures of a whole mouse brain that you see here, and then move in to study the subcellular details, such as with these neurofilaments. All this is done using the same system. Following the workflow that was introduced by Marcus, the next step requires refractive index matching for 3D imaging. The imaging solution that comes with the clearing kit is non-toxic and provide a reproducible and accurate refractive index matching that is optimized to provide a high image quality with our objective lenses. The workflow presented here enables imaging across scales, and to demonstrate, here we have a whole mouse brain that was imaged from macro at 0.6x all the way to 30x, where we reveal the small vasculatures deep within the brain at a high lateral resolution of about 500 nanometer. I would like now to go over some of the work that was done by our users who relied on our technology to answer questions that had not previously been possible to answer before. The first application here is melanoma diagnostic. This great work was done by the group of Professor Gunzer in the University of Essen. The conventional method for melanoma diagnostic involves analyzing only a sample of 20 sections out of a lymph node. This sampling leads to loss of information and inconclusive diagnosis, with about 10 to 15% false negatives, with the respective patients ending up without any therapy options. Gunzer Group transformed this procedure with the UM. They imaged and analyzed the entire lymph node, and that provided them with a much more comprehensive and reliable diagnosis. Metastasis was detected in biopsies that often gave false negatives using conventional techniques. Therefore, affected patients were provided therapy much earlier. In addition, large-scale 3D imaging allows studying the distribution and quantification of CAR T cells in solid tumors without the need to do any sectioning. And here you can visualize in the middle the tumor cells in green and CAR T cells in violet and vasculatures in orange within large solid pancreatic tumor. This pioneering work was performed by my colleague Wael Al-Rawashdi, who is transforming the immuno-oncology field with innovative imaging modalities. Last but not least, the UM can be a powerful tool for metastasis quantification, where conventional methods such as positron emission tomography often fail to detect micrometastasis and lead to incomplete assessment of therapies. With the UM, the group of Dr. Ali Erturk from Helmholtz Institute were able to detect metastasis with cellular resolution in a whole mouse body, and then evaluate therapeutic antibody targeting. This exciting work has never been done before. This 3D data set of a mouse lung is from the same study. We can fly through the lung, and in magenta, you see the tumor metastasis with sizes down to a single tumor cell. And if you inject the mouse with a therapeutic antibody that has a different dye than that of the cancer cell, this would allow you to see which of the tumors were targeted by the therapeutic antibody, and then you can proceed to quantify the metastases that were missed by this therapy approach. As you can see here, large-scale 3D imaging can transform drug discovery research. When it comes to neuroscience, the UM has been an important tool for holistic analysis of the nervous system in health and disease for the last decade. The first major application that benefits from 3D imaging workflow that I would like to highlight involves the study of Alzheimer's and Parkinson's disease, where researchers successfully revealed the localizations of amyloid plaques and dopaminergic neurons in whole mouse brain that can be used then to study the pathology and evaluate therapy for these diseases. This workflow could play a key role also in revealing neuronal connectivity in whole mouse and help identify the effect of central nervous system trauma on the peripheral nervous system. The work summarized in the middle by Dr. Ali Erturk shows the damage that was induced in the brain, how it resulted in an effect observed and measured in the thorax, in the dotted square you see in the middle. This highlights the importance of the holistic approach of the UM that can be crucial for therapy evaluation. In addition, cerebral organoid can be imaged in multicolor and high details, as you can see in the video on the right. This dataset with the organoid was prepared using the full workflow that Marcus and I presented today. A cerebral organoid, or sometimes called brain organoid, is an artificially grown in vitro miniature organ resembling the human brain. And it can be used in application ranging from tissue morphogenesis, transplantation, all the way to drug testing and evaluation. Let's have a closer look at whole brain imaging. Here's an example where whole brain imaging was used to study Parkinson's disease. The high resolution allows us to see individual dopaminergic neurons, which is necessary for later to quantify and do a quantitative analysis. Dopaminergic pathways are involved in many brain functions, and the dysfunction of these pathways may be involved in Parkinson's disease. This quantification that you see here done by Gupra using the ultramicroscope shows a loss of dopaminergic neurons in mouse models with Parkinson's disease compared to control models. Researchers did not stop here. When the pandemic was upon us, they were hard at work trying to understand the different aspects of SARS-CoV-2 infections and COVID-19 pathology. In a very recent publication, researchers demonstrated the utility of 3D imaging workflow with the ultramicroscope to investigate host-pathogen interactions of the virus at a mesoscopic scale. In this video on the left, we see a fly-through animation of individual infection sites in ferret nose 4 days after infection. Blue here represents the autofluorescence background, and green and magenta shows the infections. With the successful 3D visualization of infections, this study serves as a proof of concept for investigating important respiratory pathogens in intact tissue and organs. Although this is considered a respiratory disease, researchers recently used 3D imaging with the UM to analyze the distribution of the virus in the whole mouse brain, as you see here. The paper that is summarized on the right provides evidence for the neuroinvasive capacity of the virus. While conventional immunohistochemistry studies can still be used to study infections, none of them are able to provide a greater spatial context of the disease that can be enabled by our 3D workflow solution. To summarize, we showed you how the 3D imaging workflow with the ultramicroscope can not only transform research in immuno-oncology and neuroscience, but also it helps answer questions that were previously out of reach with conventional methods. We know that integrating a new technique into your work requires time-consuming efforts in order to get it optimized. For this reason, we provide ready-to-use reagents and a simple protocol to get your large-scale 3D imaging experiment started. And the fully automated light sheet imaging with the UM Blaze takes your imaging experiments to the next level, providing you with the ability to image across scales at unprecedented details.